In this video tutorial, I am going to discuss isoenzymes of lactate dehydrogenase, commonly known as LDH. I will cover their structure, how they are distributed in different tissues, and how they are useful in diagnosing various diseases. Along with that, I will also share a simple trick to remember which isoenzyme is found where. So let's get started. First, let's understand what lactate dehydrogenase actually does. As the name of enzyme itself suggests, it dehydrogenates lactate, means removes hydrogen from lactate and convert it into pyruvate. This hydrogen removal occurs with the help of NAD+. So NAD+ gets converted to NADH plus H+. But remember, this is a reversible reaction, meaning by exactly following this reaction in reverse direction, pyruvate can also be converted into lactate. In human body, LDH is present in virtually all the tissues. And if we consider individual cells, it is present in the cytosol fraction of the cell. Let's talk about the structure. LDH is tetramer, which means it's made up of 4 subunits. Each subunit can be one of two types. H type, here H stands for heart, or M type, here M stands for muscle. The H subunit is coded by a gene on chromosome 12, while the M subunit is coded by a gene on chromosome 11. And we know from our discussion in the previous video that when enzymes or their subunits are derived from different genes, we call them true isoenzymes. So here LDH qualifies as a true isoenzyme. One more thing about this subunit is that this H subunit is heat stable at 60 degrees Celsius, but this M subunit is heat labile, meaning it denatures easily with heat. You can easily remember this, H for H subunit and H for heat. Now, by arranging this H and M subunits in all possible combinations of 4, we get a total of 5 different isoenzymes. Let's enlist them. First combination is all 4 H subunits. We call this H4. It is completely heat stable as all are heat stable H subunits. Second, 3H and 1M subunit, that's H3M. Still heat stable as the majority are H subunit. Third is 2H and 2M, that is H2M2. This one is partially heat stable as only half are H subunits. Fourth is only 1H and 3M, that is HM3. Now it's heat labile due to majority of subunits being that heat labile M subunits. And fifth is all 4 M subunits, that is M4, completely heat labile. We can separate these isoenzymes in serum using electrophoresis. So, suppose this is the electrophoretic medium. This is the negative electrode and this is the positive electrode. The negative electrode is known as cathode and the positive electrode is known as the anode. I know this sounds confusing. We call positive ions as a cations, but the positive electrode is not called a cathode. It's actually called an anode. Similarly, negative ions are anions, but the negative electrode is not an anode. It's actually cathode. The name don't match up the way you would expect. Now, the serum sample is applied over here. Once the electrophoretic run completes, LDH is separated into 5 different bands. Now, what names should be given to these bands? By tradition, numbers are always given from anode side to the cathode side. So in this case, this band is nearest to the anode. So this gets LDH1. The next gets LDH2, this LDH3, this LDH4 and this LDH5. In other words, LDH1 is the fastest moving as it had traveled this much distance, whereas LDH5 is the slowest moving as it had traveled only this much distance. So in other words, you can also say that the fastest moving isoenzyme is always given number 1 and the slowest moving is always given the last number. Now the fact is LDH1 is actually this H4. LDH2 is actually this H3M. LDH3 is H2M2, LDH4 is HM3 and LDH5 is M4. So now I think the entire picture is clear to you. Now comes the most important part. Ideally speaking, LDH is very widely distributed enzyme in our tissues. But some tissues contain some isoenzyme forms of LDH predominantly. So which isoenzyme is predominant in which tissue? LDH1 is found predominantly in heart. LDH2 is found predominantly in the red blood cells. LDH3 is found predominantly in the brain. LDH4 is found predominantly in the liver. And LDH5 is found predominantly in the skeletal muscles. One more thing about this LDH4 and LDH5. Both are predominantly found in both the tissues, liver and skeletal muscle. But liver contains slightly higher LDH4 activity compared to LDH5, and skeletal muscles contain slightly higher LDH5 activity compared to LDH4. That's why I had written only liver over here and skeletal muscle over here. But as a second rank tissue, we can add skeletal muscle over here and liver over here. Now, many students find it difficult to remember which isoenzyme is predominant in which tissue. But I generally remember it visually by this simple trick. Can you see this number 1 in heart, number 2 in RBC, number 3 in brain, number 4 in liver, and this number 5 in skeletal muscle? If you remember by this visual trick, then you can never forget this concept. Remember, LDH is present intracellularly in the cytoplasm. In serum or plasma, which is extracellular fluid, it's non-functional enzyme, meaning it serves no function there. However, due to normal cell turnover, some intracellular LDH spills into the serum. And that's why we see low level of LDH activity present in normal serum, and that is around 125 to 220 units per liter. And this serum LDH activity is what routinely measured in clinical laboratories. Inside the cells, enzyme concentration is about 1500 to 5000 times greater than what's normally found in serum. Now, if we look at the normal serum, the distribution of various isoenzyme fraction is interesting. LDH1 makes up to 25% of total serum LDH activity. Similarly, LDH2 makes up 35%, LDH3 is 25%, LDH4 is 10%, and LDH5 is only 5%. Here, one thing to notice that LDH2 is maximum in serum. Why? Because it comes from where? Red blood cells, which have lifespan of only 120 days. This is the shortest lifespan among all tissue where LDH is present. So LDH2 more frequently spills into serum. So LDH2 fraction is highest in normal serum. Now let's talk about diagnostic importance. Isoenzymes of LDH are used in the diagnosis of heart and liver related disorders, though this has only historical significance now. In healthy individuals, the activity of LDH2 is higher than the LDH1 in serum, as we had just discussed. But in case of myocardial infarction or heart attack, heart cells get damaged, and as discussed earlier, heart cells contain predominantly LDH1. So this LDH1 comes to serum, and LDH1 becomes much greater than LDH2. This is called LDH flip pattern because the pattern is reversed as compared to normal subject. In heart attack, LDH levels start rising in 12 to 24 hours, reaches its peak level in 48 to 72 hours, and may remain elevated for 10 days. This timeline is important. Now, in case of liver diseases, as hepatocytes contain predominantly LDH4 and LDH5, increased activity of LDH4 and 5 in serum is observed in case of liver disease. However, the use of LDH in diagnosing of heart and liver disorders are mainly of historical significance now, because nowadays there are better and cheaper laboratory tests are available. The reason I had mentioned this obsolete usefulness of LDH lab test is that most biochemistry textbooks still emphasize these disorders. So I think to get a mark in your exam, you need to write this. However, you will rarely use LDH lab test to diagnose heart and liver related disorders. That's one sad truth. One more thing, in current clinical practice, serum LDH measurement is relevant only in hematological disorders like megaloblastic anemia, hemolytic anemia, and various cancers. The deep understanding of current clinical practical use of LDH is covered in my other video. You can find the link in the description and in the info button. There is one more extremely important thing. LDH activity in red blood cell is 4,000 times more than that in serum. Some popular biochemistry books mention 80 to 100 times, but that's wrong. It's actually 4,000 times. This means for estimation of LDH or its isoenzymes, the serum sample should be totally free from hemolysis. If the sample is hemolysed, that means RBCs are broken down and the contents of RBCs are transferred to serum. So you will get false high LDH activity in serum because even a tiny amount of RBC breakdown will flood the serum with LDH. So to summarize, LDH is a tetrameric enzyme with two types of subunit, H and M, giving us 5 different isoenzymes. Each isoenzyme has a specific tissue distribution, which you can remember using the simple visual trick we discussed. While LDH was historically used for diagnosing heart and liver diseases, its main clinical use today is in hematology and oncology. That's it for this video. If you have any doubts or question, feel free to drop them in the comment section below. I'll be happy to help. Thank you.